Hello, everybody. Um, this is a quick walkthrough of the Forensic DNA Lab Part 2, the PCR amplification of DNA extracted from hair. Um, so um, here's a quick overview of the polymerase chain reaction, or PCR. This has been a powerful tool that revolutionized biotechnology, and it has numerous applications, but the major ones include molecular cloning, sequencing, and forensic applications. So the idea is that today you will use the DNA sample that you've extracted last week, and you'll use primers to amplify small portions of the DNA sample. And the amplification will be done by a DNA polymerase. And the DNA polymerase will be mixed together with some buffers and nucleotides into a polymerase mix. And this reaction will happen in a PCR tube on a thermocycler. And the idea is that the thermocycler will go through several cycles of amplification, and each cycle will consist of the following. There will be an initial step where the DNA is heated to 95 to 98 degrees Celsius, which melts the DNA and separates the two DNA strands. And then this DNA the temperature is lowered to about 55 degrees, which allows the short DNA primers to bind to the DNA template at very specific sites. So we'll have primers that bind to a site on chromosome 16, and we'll have primers that bind to a site on chromosome Y. And then the temperature is raised to 72 degrees, a temperature at which this polymerase can extend the primers and make synthesize long pieces of DNA. In our case, our PCR products will be on the order of 200, 300 base pairs. And this cycle is repeated numerous times to amplify the same piece of DNA over and over. In our case, we'll have 40 cycles. So the goal of today's lab is to amplify DNA extracted from hair last week. And like I said, we'll have two sets of primers. One will amplify a 271 base pair fragment for chromosome 16, and the other set of primers will be used to amplify a 313 base pair fragment from chromosome Y. And the idea is that women and men both have chromosome 16, but only men have chromosome Y. So in addition to your DNA template that you've extracted, we will have control DNA samples, XX control DNA sample extracted from a female, in this case, Ms. Hammack, Science teacher at Oak Grove High. And we'll have XY control DNA extracted from your teacher, Dylan. So the idea is that when we PCR XY DNA, it will have chromosome 16. So you'll have a product for chromosome 16, and you'll also have a product for chromosome Y. But when we'll PCR DNA from a woman, we'll get the we'll get the product for chromosome 16, but we'll not get a product from chromosome Y because women don't have chromosome Y. And hopefully, if you've extracted your DNA correctly, we'll get products for your DNA. And the only difference is that the control DNA was extracted using a different method, which gives a lot more DNA. So we know that the control DNA will give a very robust product on a gel. Okay, so let's review today's PCR protocol. So I've made this table that you will have to follow very carefully to make sure that you assemble the correct reactions. And it's absolutely critical that you keep track of which reagents you already added to which tube. And please take it slow and make sure that you don't confuse tubes from which you're pipetting or into which you're pipetting. And so the first thing you'll do is you'll have 6 PCR tubes for 6 reactions, and you'll label these reactions number 1, number 2, number 3, number 4, number 5, number 6. The tubes are very small, so you can't write full text, so you just write numbers. And these reactions will be control DNA, XX DNA with primers for chromosome 16, control DNA, again, XX with primers for chromosome Y. And then you'll have two reactions with XY DNA for, again, chromosome 16 and chromosome Y primers. And finally, you'll have your DNA with chromosome 16 or chromosome Y primers. And in each row, we have individual reagents, for example, chromosome 16 primer mix. You will add this to reactions 1, 3, and 5. You will not add it to reactions 2, 4, and 6. And then you will pipette chromosome Y primers to reactions 2, 4, and 6. After that, you'll pipette the control DNA template, the XX DNA, to reactions 1 and 2, the XY DNA to reactions 3 and 4, and then your DNA to reactions 5 and 6. Oops. And when all of this is done, then you will pipette the polymerase mix into all the reactions. So the polymerase is called Q5 polymerase. And the important thing here is you will get the Q5 polymerase only when you're ready to pipette it. So at the very end of the lab, because this enzyme is stored at -20, and you will ask the teacher when you're ready to pipette it. And finally, after you've pipetted the polymerase, you'll pipette mix 10, 15 times to make sure that the reaction is fully mixed. There will be a quick centrifugation step to make sure all the liquid goes to the bottom of the tube, and then you can run the reaction on the thermocycler. Okay, so the first thing you will do is you will prepare your bench, make sure you have your protocol, make sure you have a tube rack, a pen, your pipette, your tips. If you have ice, it's nice to keep things on ice. If you don't have ice, that's okay. You can assemble the reactions at room temperature. And okay, so the first thing you do when you start pipetting, double check that all your tubes are labeled. One of the things would be nice to do is to label the side of the tube with your team initials, because every team will have 1, 2, 3, 4, 5, 6, and you need to make sure that you know which team the tube came from. So in this case, Dylan did the reaction using an ice bucket, which you may or may not have, and his primers are labeled a little bit different, but 3, 4 is his chromosome 16. Then you have chromosome 1 primers. Then you have the DNA extracted from hair, control DNA, and then he had some water. In this lab, you don't need water. Everything has been pre-aliquoted. Okay, so his protocol is a little bit different, but that's okay. This video is meant to show you the key steps in the process. Okay. So the first thing you will pipette are the primer mixes, and you will pipette 2 microliters of primer mix per reaction. So keep in mind that you're pipetting chromosome 16 primers into reactions 1, 3, and 5, and chromosome Y primers into reactions 2, 4, and 6. And every time you pipette something, you pay attention to the tube. So what are you pipetting, and what tube are you pipetting into? So let's see, Dylan, he's pipetting primers for chromosome 16, taking 2 microliters. And he's pipetting it into reaction number 1. After you pipette, you eject the tip and you close the tube to make sure that we don't contaminate it with anything else. And then you cross what you have already pipetted. And something else that Dylan is doing here is he is moving the tube to the side as another way of indicating that he's already pipetting that step, pipetted that step. And now the same primer mixture goes into tube number 3. So this time he pipetted all the liquid to the bottom of the tube. And again, critical to keep track of what you have pipetted. And the same primer mixture goes into tube number 5. So at this point, he's done pipetting the first primer mix. Great. So after pipetting chromosome 16 primers and chromosome Y primers, you can move on to pipette the template DNA. And the template DNA, you will pipette 4 microliters of DNA per reaction. So the XX control DNA goes into reactions 1 and 2, XY control DNA goes into reactions 3 and 4, and the DNA that you extracted goes into reactions 5 and 6. And one of the things that you may encounter is that sometimes the liquid in the tube is not all at the bottom. Some of it is on the side of the tube. And if that's the case, what you can do is to quickly centrifuge the tubes to get the liquid to the bottom. And Dylan here is using a mini centrifuge, which is a little bit bigger. What you will probably have at the bench is one of these microcentrifuges, which has already adapters for PCR tubes. So load your tubes in here, make sure they're balanced. So you have 3 tubes into one side of the rotor and 3 tubes on the other side. Do a quick spin and you'll be done. Great. So now your protocol should look something like this, where you have pipetted chromosome 16 primers, chromosome 1 Y primers, all the DNA templates. You've crossed out all the things you've pipetted, and now you're ready to pipette the polymerase mix. And like I said in the previous slide, make sure all the liquid is at the bottom, because you will need to mix the polymerase with the primer and the DNA that you've previously pipetted. Right, so you will need 6 microliters of polymerase per reaction. And you will get your polymerase from the teacher. In this case, in your case, it will be labeled with the letters Q5, and it may be frozen when you get it. So thaw it in your fingers. You can take your 20 microliter pipette, set it to 20 microliters, and pipette the polymerase mixture up and down 10 times after you thaw it, to make sure that it's um, fully mixed. Sometimes there might be small ice crystals, or maybe salt crystals, sort of precipitated salt. You want to make sure that it's homogenized, and then you can pipette it into your reactions. Right, so you will need 6 microliters per reaction. That's what Dylan is doing here. So after you pipette the polymerase, you'll mix up and down 10 to 15 times. If you do not mix the PCR reaction, it will be very inefficient. You'll get a lot less DNA, or maybe you'll get no DNA. And if you have an ice bucket, place the tube on ice. If not, you can leave it at room temperature. It will only take you a few minutes to assemble all the reactions. So after you've assembled all the reactions, your protocol should look something like this, where everything is crossed out. And you are ready to load your reactions into the thermocycler. But before you do that, give your tubes another quick spin. I'm going to fast forward here. Quick spin. And take it over to the thermocycler. And you can take your time to review the protocol. So What we have here is initial incubation, 30 seconds at 98 degrees. Another incubation of 20 seconds at 98 degrees, which melts the DNA. 20-second incubation at 55 degrees. This is the primer annealing step. Then a 30-second incubation at 72 degrees, which is a primer extension step. It's hard to see here, but um, there's a go to step number 2 and repeat 40 times. So this is where the amplification happens. And finally, there's another 1 minute incubation at 72 degrees and indefinite incubation at 4 degrees. And the teacher will collect the samples from the thermocycler once the reaction is done, and we'll put them at in the freezer at -20. Right, so after you review the protocol, you can load your samples. The order in which you load the samples doesn't really matter. But it's important that the tubes are closed tightly. And when you close the lid, you can turn the knob to make sure that the lid is nice and tight. Do not turn it too hard because that will crush the tubes. And when you're ready, you can press run, and you will know that the protocol is running because the timer is running. And this reaction has 2 hours and 18 minutes remaining, so it will finish after the lab is over, after your class is over. Okay, so this is the end of the PCR amplification lab. In the next lab, you will analyze the results of today's experiment by running your PCR reactions on a gel. And good luck, and see you next week.